So, in case you don't know, from the University of Strathclyde, which is in Scotland, which is this red dot here, but currently the map of Scotland looks like this. So it's quite nice to be in Orlando, where it's a tad warmer than what it is in Scotland like right now, but I don't actually know if I'm going to manage to get home or not, but that's tomorrow's problem. And just showing you another picture of how deep the snow currently is. One of my PhD students had posted this, and yeah, it's quite bad for Scotland. So anyway, the sunshine's a nice change to the, well, although I'm missing 3 snow days, which might have been quite nice. So anyway, Alison, my colleague's nodding her head with me there. Okay, so my areas of research involve using Raman spectroscopy and specifically surface enhanced, although we do some Raman imaging as well. The areas we're particularly interested in is the overall area, I guess, is bioanalysis, with a particular emphasis on sensitivity and multiplexing, so as to be able to detect low concentrations, but also multiple analytes in the one sample. We've done a lot of work previously in molecular diagnostics, detection of DNA sequences that we'll talk a bit about today, as well as developing different protein assays using functionalized nanoparticles. We've done some work in patterning, particularly writing down molecules or nanoparticles on surfaces. So, for example, to look at stem cell differentiation on surfaces based on the stiffness and the spacing of the molecules in between. We're doing a lot of work in imaging, so I'm not going to speak about that today. So, both looking at nanoparticles inside biological systems and imaging, a bit of drug delivery, and we're doing some work on SERS as well, which Bavia spoke about earlier, so I'm not going to speak about SERS today. And we also do some small molecule detection, so drugs, explosive molecules as well, various different approaches, but all generally using Raman and surface enhanced Raman. So Bavia's already given a nice introduction already earlier to SERS. So obviously we're talking about absorption of molecules onto rough and metal surfaces, and we tend to mainly focus on nanoparticles, so silver and gold. Predominantly, historically, we've used a lot of silver because we get strong enhancements for silver surfaces, but now that we're starting to look into more kind of in vivo or moving into human animal models, that we're tend to make more redshifted nanoparticles, so gold. We like to, where possible, use the resonance enhancement as well. So if we tune our excitation wavelength to the analyte molecule, we get greater enhancement. So if we tune our excitation wavelength to the colour, the absorbance of our analyte, then we get much greater enhancements in the Raman scattering back. We've done a lot of work using commercially available fluorophores because there's such a suite of them out there that we can tune our fluorophore absorbances to our excitation wavelengths. It means we can also tune into existing labelling chemistry, so we don't need to develop our own. So we've done a lot of work in direct detection of DNA sequences labelled with commercially available fluorophores. So as well as obviously getting your fluorescence spectrum, if you absorb it onto the surface, you also get the SERS spectrum, and our nanoparticle tends to quench some of that fluorescence, allowing us to get our Raman rather than just the fluorescence response. So I mentioned we're particularly interested in multiplexing. So what do I mean by this? Of having multiple either individual analytes that we can absorb onto the surface of one nanoparticle, for example, or using different coding, so different dye labels on the surface of different nanoparticles that are then coated with some biorecognition molecules, such as an aptamer, an antibody DNA sequence that we then use to target something within the system. So we get our individual spectra. We then, if we have these all in one system, we get our multiplex spectra, and we're interested in, well, how do we work out back the way what the individual analytes were present? How do we deconvolute that data within those systems? So we'll talk a bit about that. So I guess there's 2 main approaches to doing SERS using nanoparticles. So there's direct absorption, where you just take your nanoparticles, absorb your analyte onto the surface and detect it directly. Or you use the nanoparticle as a label, essentially where you functionalize the surface and use the entire nanoparticle as a label to go and sense for something. So I'll talk about 2 approaches. But I guess fundamental to any of these approaches that you use is that you really need to control and understand your nanoparticle surface to get what you want onto that surface. So we've done a lot of work on the detection of DNA sequences. So when you make nanoparticles, they're well, mainly most synthesis methods, you end up with them being highly negatively charged on the surface. So obviously if you have a positively charged analyte, that's fine because you can electrostatically attach them to the surface, but if they're negatively charged, you need to think about your surface chemistry and how you're going to get them onto the surface. So for DNA we've used spermine, which is a polyamine. So one, it coats the nanoparticles with a positive charge, but also aggregates the nanoparticles. So when you go from isolated nanoparticles to aggregated nanoparticles, you also get an increase in the SERS response. But what this does is allow us to bring our DNA, which is negative because of negatively charged backbone, down onto the surface. We don't see much SERS from the DNA sequence itself because we've got a big resonant label on here, so we're looking at the response from the label rather than the DNA itself. This is a horrible table, so just ignore most of it, but these are different DNA sequences with different labels attached with the absorbance of these labels. But just what I want to highlight is I mentioned that we use the resonance effect where we tune our excitation wavelength to the absorbance of the molecule. And when we do this, so if we use 514 with this TET label, which absorbs at 521, so these are in resonance, you see we get detection limits on the order of 10 to the minus 11 molar. However, if we move away from resonance and use 63, we lose 2 orders of magnitude in sensitivity, or should I say, when we're in resonance, we gain 2 orders of magnitude in sensitivity. So if we get these conditions right, we can get really sensitive detection. So obviously, DNA is not naturally labelled, so how do we have to create assays to detect it. So one assay that we developed is using this enzyme, lambda exonuclease. So essentially this has a toroidal structure where one end, the hole in the enzyme essentially is bigger at one end than the other end, so it allows double-stranded DNA in and then digests one strand and lets single-stranded DNA out the other side. It also requires a 5' phosphate group for recognition. So the assay we developed, so this green squiggle is our target DNA. If we design one probe that is complementary to half of this and it's got a biotin label on it, I'll tell you why in a second. And this is our SERS probe, so it has complementary to the other half of the DNA sequence, so it's a sandwich probe, has our SERS label on here, and it has a 5' phosphate group. The 5' phosphate group is a recognition site for the enzyme, so that has to be present for the enzyme to digest. So when our target's present, this will all hybridize. So the reason we put that biotin group on here is so that we can add magnetically coated streptavidin beads that allow us to wash away any excess probe. Because if we have any excess signal in there, then we'll get a background response. So once all this occurs, we add our enzyme, we then digest, and then we get our SERS response and look in here. So basically to get a SERS response, we needed all the components to hybridize the correct DNA sequence to be present there. So I said we're interested in use multiplex detection. So the reason is we want to be able to detect more things in one sample essentially. And the example that we're using for this is for bacterial meningitis pathogens. So these are normally detected in CFS. Most people that have meningitis, unfortunately, are newborn children who have not yet been vaccinated against it. So obviously the CFS sample, one, it's highly traumatic to take a CF sample from a newborn or small child, but the sample is obviously very small. So it's a good example of where we have a very small sample and want to get a lot of information back from it. So we were looking at 3 different pathogens. Obviously those 3 pathogens wouldn't actually be present at the same time, otherwise I think the patient would not live long enough for you to take a CFS sample of them. But using this assay we designed 3 probes, each of which are complementary to each of the different pathogens. So this one labelled with TAMRA, FAM and Cy3, so they've all got unique SERS spectra. We can see our controls when there's no target present or there's non-complementary DNA present, we don't get a signal. So it works nicely. Oh, there's something wrong with that slide. It all triggers. So we also got nice quantitative detection, which we necessarily didn't actually think would be the case because the assay requires an enzyme digestion, hybridization. There's quite a few steps in there that we thought that it might not actually give us a linear quantitative response, but it does, and we get very sensitive detection, and we can do this for each of the single pathogen species. Now, as I said, we're interested in multiplex, so in this case all three would not be present, but in other conditions you might have multiple bacteria present, or for example in fungal infections it's actually quite common to have multiple infections. But also what we're interested in is also the different levels. So yes, we don't want to just say, well, there's 3 to 1 of them present, but we want to be able to quantify how much of each is present. So if you maybe have a low-lying bacterial infection where one's present, but obviously another one's at a much higher level, which is what the actual issue is. So we designed all our probes that they could be easily seen. So we picked labels for them all that could be easily seen within the multiplex. So if you follow this up, there's in the multiplex where all three are present, there's 3 unique peaks, if you like, for each of the species. However, that allows us to see it visually, but as we move to lower levels and different concentrations of each, it becomes harder. So obviously we wanted to use, multivariate analysis. So we can see that using multivariate analysis we can separate. This is a multiplex spectra and these are the singplex. What you do see is the multiplex is closer to the FAM spectrum, and Bavia mentioned this earlier, is that, this is a surface-based technique, so one of the species has a stronger affinity to the surface or a stronger enhancement, then it's going to dominate that multiplex a bit more. So we do see that within the FAM, that there's more FAM character within the multiplex, if you want to put it that way, but we can still separate them easily. As I said, we wanted to try and quantify this within the multiplex. So this is in collaboration with Roy Goodacre at the University of Manchester. So we set up a system where we have all 3 of them present, but they're all present at varying concentrations within that multiplex. So there's a matrix of 66 samples. There's 10 different concentrations of each of the labels within the multiplex, all at varying amounts at different times. So 66 samples, and we did 5 replicates of this. So poor Kirsten did all this work in one day, a very, very long day in the lab doing all these replicates. But we got really nice data back. So these are the PCA plots. They're separated into each of the different components. And what you see, basically the size of the spot is related to the concentration of the sample. So if we look at that dot here, that's the same sample. It's got a low concentration of, I've just realised that the labels are missing there. You'll know it's there. The FAM probe, high concentration of the TAMRA and a low concentration of the CIT, and that relates all the way through. But the nice shape of it shows basically we can quantify. So using half of the data for training, another half for testing the set, we get this really nice linear concentration dependence. So we can pull out the curve, the concentration curve for each of the analytes, but always remember that these are all within the mixture as well, so we can, no matter what other levels of the other analytes are present, we can quantify within that. So it was really nice to pull this out. We then did some blind samples essentially, so we ran the samples and prepared them, and we sent the data down to Manchester, so they didn't actually know what the concentrations of the sample were. So these are the actual values and these are the predicted values. So the correlation is quite good as you look across. What you see, the main issue here is we get into a bit of trouble with that FAM probe again, and we know this is because it's dominating the spectra. So actually this one is really bad. I shouldn't point out the the bad parts of the data rather than the good parts. But you'll just see that coming through in this particular sample. For example, there was 0 present, but it's shown up here, and there's a slight over-prediction here, and it is because the model is slightly over-predicting. So we have done this with some clinical samples from Glasgow Royal Infirmary as well, so that it worked quite nicely, but I'm not to show you all that data. I need to watch my time. So another system that we're looking at is HRP TMB. So TMB is a substrate that's commonly used for ELISAs, so for enzyme-based assays. So this parent diene, in the presence of HRP and peroxide, turns over the substrate and it produces this blue coloured electron oxidation product that's a mixture of this radical cation and the charge transfer complex. So it absorbs 650 nanometers. Normally in ELISAs they add acid and push it to the yellow product, which absorbs at 450 and measures that because it essentially finishes the reaction. So we were like, this is a really nice substrate. The yellow product has an absorbance at 650, so it could be in resonance with our 633 laser excitation wavelength, but actually originally we thought this, the 450 peak, 512 will get some nice resonance contributions here. What we see is we do get really nice Raman signals, so the yellow product at 514, which is in resonance, and then the 633 where it's off resonance, we get nice spectra. When we move to 514, which is off resonance for the charge transfer, and then 633, which is on resonance for the transfer, what we actually nicely see is that these resonant Raman peaks actually start to appear that are not present in the Raman spectra. So you can see these are much more intense, so already we're getting nice Raman itself anyway, but now we have this resonance Raman contribution, and these peaks start to dominate and you don't see any of the Raman anymore at lower concentrations. We've got a really nice resonance Raman system. So using standard ELISA approach, putting antibodies down on microtiter plate, adding our target protein, adding our detection antibodies, so these are labelled with HRP. The HRP turns over that TMB substrate to form the blue product that they're going to detect using resonance Raman. We're not going to use that stock product. So what you can quite nicely see here, oops, go back one, is this is the colorimetric standard response, and this is the resonance Raman. So basically we're getting a good order, so magnitude and sensitivity here, because you see it starts to tail off quite quickly with the colorimetric and the resonance here. So this is resonance. This is not even being fancy about it. We're not doing SERS, so we were like, that brilliant, let's do SERS, it's going to be way much better than this, but we're already beating colorimetric. But we also did some work. The benefit of even just doing normal Raman here is that we can use portable instruments. Because we actually did a collaboration with LGC, some work here that we published recently on cortisol detection. So they were interested in developing cortisol testing in athletes, trackside essentially, and they were using a chemiluminescence response, and the portable instrument that they have in the market is not a portable instrument. It's kind of back of the car type instrument. So they wanted to know whether they could do it more sensitively, easier, and we just essentially swapped the TMB into the assay. And did use the portable detector, and lo and behold, we're still getting the same detection limit. So it's not actually doing anything fancy. We haven't really changed the assay, but it's actually just changing your detection technique to get a sensitivity. I detract. But of course, yeah, we wanted to then do SERS. We're going to go, this is going to be great. We're going to have even better sensitivity. So if you look at this, the top one's the resonance Raman data, nice linear response. This is the SERS data. So when we put nanoparticles in there, you can see these are plotted on the same scale. So you can see when we put nanoparticles in, we have got enhancement. We've got much higher intensity here, but this went to hell, basically. Our linearity, our response is no longer reproducible. But what you notice here at this point is our blank is not blank, which is the main issue that's skewing this off. So Stacey was doing this work and we just went, oh, something wrong here, don't understand it. But then a few years later, Christy was working on a similar system. We found this again, and what we discovered after quite a lot of pain, it was a different system Christy was working on as well, is that the nanoparticles themselves have a catalytic effect. So this has been published before for iron, and a little bit of work on gold, but obviously if you're not looking at that literature in relation to your system, but no one had published it before, and the fact that silver did this as well, so it never come up. So essentially the peroxide is broken down by the catalytic effect of the nanoparticle, which then turns over that substrate and so you get a colour response. So none of our blanks were blank because the nanoparticles were turning over the substrate. I just show you, if we remove anything from it, we don't get a signal, but it's when the nanoparticles, the TMB and the peroxide are present, we get a strong SERS response. And if you vary the peroxide concentration, you get a linear response, so you can use it as a peroxide detector if you want to. But we wanted to use this in an assay format, so how can we use this to our advantage rather than it being a very bad interference in our reaction? Well, how can we actually use it to our advantage? So we wanted to develop a proteinizer assay based on this, so what we now call SLISA, which is, Shan hates this, but I've managed to turn her around saying that it is actually quite a good term. So instead of ELISA, enzyme linked immunoassay, we're now calling it silver linked immunoassay. See what we did there? So if we write down our capture antibody, in this case we were looking at CRP detection because we've got a programme on cardiovascular disease, so we're interested in, and sepsis, so we're interested in looking at inflammatory markers. So essentially now we have a nanoparticle, we functionalise it with the antibody, so the antibody is a recognition element, but we no longer need an HRP enzyme on there because essentially our nanoparticle is going to act as our enzyme. So we have our kind of enzyme and antibody in one system. When we add our TMB substrate, if this binds, then the nanoparticle turns over that substrate and we get a colour. The substrate then goes down onto the surface, so rather than being free in solution, it goes onto the surface and gives us a SERS response. So after quite a lot of pain, anyone developing a surface-based assay, there's a lot of issues with blocking and non-specific absorption, but essentially this is a spot within the spectrum, on the surface. This is where the nanoparticles are present. This is where they're not present, using dark fields, so that's quite a nice discrimination. However, the Raman's more sensitive, so we get an off spot signal background, but by changing the blocking conditions, she got a really nice off-on response so that we're only seeing signal when the target's present. Oh, I played with that, shouldn't have done that. So you can see there's a nice concentration dependence as well there, where we're getting really nice limits of detection down at 3 picogram per mil. So we're looking at different ways to exploit this for different biomarkers. CRP is quite nice to use, but that's because it's quite a nice system. The antibodies for it are quite good, but we want to start looking at other molecules as well. So in the last part, another approach where we're looking at functionalized nanoparticles is in bacteria detection. So obviously bacteria detection is of interest both clinically but also in the food processing area as well. I imagine you all understand why we don't want bacteria in our food. But the strange one that's come out of this is actually a collaboration in the food industry from some of the initial research that we've done here. And it's not actually the bacteria in the end chilled shelf food that they're actually interested in. They're actually interested in the swabbing of the production lines for their cleaning verification to make sure that they're actually getting rid of any bacteria in the food production areas. So in this approach, we take metal nanoparticle, magnetic nanoparticle that we functionalise with gold. So we've got a magnetic core, silver coating on the surface, and in this case we functionalise the surface with a lectin. So we use PEG surface layer and then EDC couple the lectin onto the surface. The reason we're interested in lectins are carbohydrate binding proteins. So there's carbohydrates present on bacterial cell walls and also cells. So this step is a non-specific step, so the lectin will bind to multiple bacteria species, so it won't bind to one particular bacteria. But that's okay because this is our non-specific step, so we can use this to bind to our bacteria, pull it down. Then we add a SERS nanoparticle that's specific to the bacteria. In this case it's got an antibody that's specific, bacterial specific, and also a Raman label that will give us our SERS response. So we can pull this down, wash everything away, and we get our SERS response. But the reason we're interested or it's simpler to have that non-specific capture step, is that if we can pull down multiple bacteria, food samples have lots of different bacteria in them, even if they're not at harmful levels. There are multiple bacteria present in most food samples. So the CONE can be used to magnetically pull down multiple bacteria. We then add a mixture of functionalized nanoparticles. So we have a specific antibody to one bacteria and a specific SERS code, and again these will bind specifically to the bacteria in this, and then we get a multiplex spectrum that we can deconvolute to work out which ones were present. So in the initial work we've been looking at Staph aureus, E. coli and Salmonella for the triplets. So first of all, we made the nanoparticles. We need to check that they do actually bind to the surface of the bacteria. So this is a 2D Raman SERS image of a single bacteria, so an E. coli, and you can see here, this is a dark field image that the nanoparticles have bound, and we get a nice SERS image showing that our nanoparticles are actually bound specifically to the bacteria. We do actually find that the nanoparticles tend to bind at the tips of the bacteria. We don't know if they're binding more to the flagella at the end. This is something that we need to think about a little bit more and look into, but I don't know if it changes what we get back, but it's just more perhaps interesting. So we designed, these are the SERS labels that we use for each of the different antibodies and the targets, and so you can see we get a nice SERS response for each of the targets. We do still get a bit of a background signal because it is a magnetic separation, so we'll need to look at how we can wash away everything so that we don't get anything, and there will be a certain amount of non-specific binding between the different bacteria species, but we do get a nice strong response and a background that we can control because we can monitor it. So we've done concentration studies in the singplex so far, and you see this is a little bit blurry, but down here this is the control, so this is our background signal, and you can see this is 10 CFU, so 10 bacteria, and at each case we can differentiate 10. I think we can go lower. You can see the sensitivities varied slightly between the different bacteria. We don't know if that's because our labels haven't been optimised yet. If we can optimise those washing steps, I think we can go lower. And when we actually put this out for review, quite a nice reviewer actually pointed out something positive, going, well, you're detecting 10 bacteria in X volume at the moment, so actually if you reduce that volume, for what you're actually doing, you might actually only be detecting one bacteria actually, in your measurement, because you're not capturing them all. Because of the volumes that you're using, which is a good point to make. So we need to think about how we can, there's parameters we can optimise to get down lower. So we've done this in the multiplex. So these are the singplex spectra, and this is the multiplex. Again, this is something that we need to tune again, because I think the malachite green within this matrix is dominating the multiplex, the triplex a little bit, so this is where we need to tune what labels that we use and get everything right. But we can discriminate each of them being present at the moment, and we can use PCA, and we can pull them all out. So this is the multiplex and this is the singplex spectra, so we can differentiate them. So now, we're going to try and push this down, try and push the sensitivity, and also push the multiplexing capability. So can we quantify within the multiplex? This system's more challenging to quantify. So in the DNA example, we were directly absorbing the DNA onto the surface, so we can control that surface chemistry and the space on the surface. When you make functionalized nanoparticles, then it's harder to control the exact dye loading and signal that you get from every individual nanoparticle response. So it becomes more complicated to quantify, but that's where we're kind of going next to try and quantify this system. And that's been finished. So basically just wanted to highlight the work. Haven't talked about any of the imaging work that we're doing today, but we're using different approaches to try and detect various biomolecules, and bacteria is particularly something that I'm looking at the moment, but we have programs looking at sepsis, cardiovascular disease and cancer as well. The multiplexing is a big driver for us, trying to get more information back per test when these are obviously biologically significant samples and also potentially low volume, low throughput samples of can we get more information back to exploit the benefits of SERS. We're also kind of moving as well as trying to portabilize these assays. We are using, like for example, the little CBEX that I showed, some of these portable instruments. But what we're not very good at is the engineering part of it. Of going, we've got our instrument, we've got our assay. How do we get them to how do we do that magnetic separation and in that context as well? So yeah, we need to be better at the engineering parts of it. We'll make it work one day, but that's where we are. So I'd just like to acknowledge, I think I highlighted most of the people who did the work as we were going through. Obviously I collaborate a lot with Duncan Graham at the University of Strathclyde, and a lot of the work I've been doing in the multiplexes in collaboration with Roy at Manchester. We also have a strong collaboration with DSTL in the UK, so Neil, we call Uncle Neil. He supplies us with a lot of the portable instrumentation and collaboration on that side of things with him. And I'd just like to thank the group, and this was a sunny day in Glasgow, of which there's not many last summer. Okay, any questions?